So making a subassembly is another way to concentrate all of your viral proteins that you need to use to build a particle. So let's look at a couple of ways to make subassemblies. One way is that you can make individual proteins and they will assemble. Remember, the structural proteins of viruses have built within them the information to interact with each other. They'll fold properly, and then they will interact and form the structures. That's been selected by many years of evolution. So here are two examples of assembly from individual protein molecules. We have SV40, the polyomavirus, where the subunit of the capsid, remember these are icosahedral capsids that we're building here. Here is a pentamer made up of 5 copies of VP1 and a copy of VP2/VP3, the green protein in the middle. And these are simply translated as independent proteins. VP1 is made on its own and it folds up into this form that's eventually going to be part of the pentamer, and VP2/3 is also made independently. And they associate to form pentamers, which then go on to form the capsid. So this is what I mean by a subassembly. It's not the whole capsid, but it's more than just one of the proteins. Here is adenovirus, just another example of assembly from individual protein molecules. Here we're making a penton with a fiber attached to it, and these are at each of the 5-fold axes of symmetry. It's what give adenovirus that satellite type look. The fiber is made from one protein that's translated then trimerizes. The penton base is made from a single protein, again, that associates, 5 of them associate to form the penton base, and then the fiber and the penton base associate. So again, you make individual polypeptides, they fold properly, and then they assemble with each other to form a subassembly. And again, you can imagine that this happens better when you have a lot of these proteins in one area. Okay, so that's making individual proteins. Not all viruses do that. Other viruses make precursors. Remember, the polyprotein strategy is a good way of making several proteins from one mRNA. We have this restriction in eukaryotic cells, one mRNA, one protein. One way around it is the polyprotein. So here's poliovirus, which makes a very long polyprotein spanning the whole genome and then processes it with proteases. And here we're looking at the way the capsid protein is formed. So we translate what is the first third of the genome, essentially called the P1 region, and that encodes the 4 capsid proteins, VP1, 2, 3, and 4. So these are made, they're released from the polyprotein as soon as the right protease is made that can clip it and release it. And when it's released, it then folds properly. Here, folded P1. See, VP1, 2, and 3 begin to associate in the structure that will eventually make its way into the virion. Then the loops are cut by the protease again, and that gives us the final structural unit, which will then get incorporated into virions, as we'll see later. So that's just a polyprotein strategy, again, where the proteins are being cleaved by viral proteases. In some cases, we need chaperones. That was on an earlier slide. The virus uses proteins from the cell, and one of them are chaperones that help maintain the proper folding of the viral proteins. Now, these can be cellular chaperones or viral, and here we're showing an example of a viral chaperone. That is, the hexon protein is made by translation of the appropriate mRNA, and then a viral chaperone, the L4 100 kilodalton protein, associates into a trimer to form the hexon trimer that will get put into the virion. So those are just a couple of ways of making sub-assemblies. Now, when viruses do assemble, they typically follow very specific steps. You can almost look at it as a assembly line for a car, and things are added as the growing car moves down the line. So here's an example of that kind of sequential assembly for poliovirus. So this is actually the entire cycle of infection, but what we should focus on here is the translation of the polyprotein from the viral mRNA. We just looked at this in some detail. The P1 protein is made, it's cleaved, it forms a structural unit. This is called a 5S structural unit because in the old days we used to measure big protein assemblies by how fast they move through a sucrose gradient. And the measurement of that speed was an S unit, a Svedberg unit. So this has stuck even though few people do these gradients anymore. So this is a 5s structural unit. And then 5 of these associate to form a pentamer. All right? Pentamer is nothing more than 5 of these 5s structural units. And again, the information for assembly into a pentamer is built into the 5s. When there are enough of these in the same area of the cell, they will spontaneously assemble into a pentamer. The pentamers then join with genomic RNA, and the RNA, of course, is made by replication in a process that we talked about some time ago. And this is, of course, happening later in infection. And now you have a capsid formed by putting 12 pentamers together with the genomic RNA. And now you have a complete virion, which is almost ready to go, except it needs another cleavage, and that is the final one between the last 2 capsid proteins, VP4 and VP2. And finally, it's an infectious particle, and it gets released from the cell. So that's an example of sequential assembly. You make a series of structural intermediates that proceed to the virion. So the assembly line is an important concept because it assures that the virions you make in the end are right, they're correct. You know, if you have a car going down an assembly line and you miss the door, at some point someone notices it, and I don't know how assembly lines for cars work, but won't reach the end of the line, okay? So it's the same idea with a viral assembly line. And this is illustrated here for a bacteriophage. So this is a typical tailed bacteriophage that we've only briefly talked about in this course. Has an icosahedral head, a helical neck, and some tail fibers to help it attach to cells. And this illustrates how this is built by an assembly line process. So for example, the first thing you do is you make the head, the icosahedral head, by a number of proteins. All these are given different numbers here, gene numbers that are involved in them, and that would be made by the same sort of process that the polio capsid is made by. But that's not the entire virion. You have to make this neck, which is done by assembling the base plate and this tube in the middle and finally wrapping other proteins around it. And then only when this is properly made can it join with the icosahedral head. And in fact, it wouldn't even get to this point if it hadn't gone through each of these steps properly. Same thing with the icosahedral head. If something goes wrong, it's never going to join up with the neck. And even the tail fibers go through a series of assembly steps of various sorts. And again, they're sequential. If one doesn't happen, the next one doesn't happen after that. So only good fibers get joined up in the end. So this gives you an orderly process that assures some quality control, that the viruses you build, at least in terms of them fitting together, are right. You don't make a bad virus. You don't incorporate a bad neck, for example, because it doesn't reach the end of the process. So let's talk about how a couple of other viruses are made. And especially with bigger ones, it's not so easy to have spontaneously assembling capsids. And here's an example for a herpes virus. Now, you may remember that these viruses, they have large double-stranded DNA genomes, and they replicate their genomes in the nucleus. And in fact, replication and putting the DNA into the capsid is tightly linked. So the assembly occurs in the nucleus as well. So all the proteins that you need to build the virion with, they get made in the cytoplasm. So here's the nuclear pore here. Cytoplasm is on the outside, on the left of this slide. And here in the middle, the main part of the slide is the nucleus. All the proteins that are going to be building this capsid, which you see here being assembled, have to be translated into the cytoplasm. They're shipped into the nucleus, so each of them has a nuclear localization signal. You can see there are lots of them. There's pentamers and hexamers and triplexes and VP24, 21, etc. These get shipped in the nucleus, and they form what's called a procapsid. Now, this is a very large virion, 2,000 angstroms in diameter, something like that. So it doesn't assemble well on its own. It needs a chaperone, essentially. And the chaperone is really in the form of a scaffold. So you can see in the middle here, as this procapsid is formed from pentamers and hexamers, there is a protein scaffold. So the protein scaffold assembles, and then the capsid forms around it. So this is like a scaffold on a building, which is typically on the outside, but this one is on the inside of the virus particle. Now, of course, you don't want the scaffold to remain because you need to put DNA inside there. And in a building, you just take the scaffold off the outside. But here, there's no way to get the scaffold out. So cleverly, one of the proteins that's part of the scaffold is actually a viral protease. And at some point in the assembly, it gets activated and digests away the scaffold. And what you're left with are remnants that don't matter. And then the genomic the genomic DNA is put in to this capsid, and it goes in through the portal. Remember, these virions have a single portal at one of the five-fold axes of symmetry. You can see it right there. It's called UL6 portal, and the DNA is put in through that. We'll look at that process a bit later. So this is a very clever way to make a big capsid, where you do need to have some support until it's fully assembled and can stand on its own, and probably the DNA contributes to that stability. So this whole idea of sequential assembly is also part of adenovirus's assembly line. Here, this virus doesn't need an internal scaffold, although it does need chaperones to assist in folding. We showed you an example of a viral chaperone that helped the hexon to trimerize. But here are the assembly steps of adenovirus in the nucleus. Remember, this is also a DNA-containing virus. It replicates its DNA in the nucleus, and it also assembles the virions there as well. So all the structural proteins, again, get made in the cytoplasm. The hexons, the pentons, the fibers, everything gets made in the cytoplasm. They get put into the nucleus. And then they go through a series of steps to form the virion. Now, we're not quite sure how adenovirus actually assembles. There are actually two models here, and this illustrates two general ways that assembly can proceed. So we've talked mainly about this sequential model for assembly, where you build things in a series of steps and make sure that they're all right, and then in the end you have a virion. So one model for adeno assembly is that you sequentially assemble first an empty capsid, and then the DNA is threaded into it till you get the virion. Starts as a young virion, and then finally it matures as the result of proteases to form the intact virion. So a series of assembly steps. You make a shell, you put the DNA in, then you mature it. There are a school of experimentalists who believe that this is not the way it happens, but rather the DNA and the proteins all come together at once. There's not a sequential series of events. And this is called concerted assembly because most of the assembly happens at one step. It's not a series of individual assembly steps. So which one is right, we don't know right now, but this does illustrate two ways that you can make a virus particle: sequential versus concerted. Now, just a few words on the need for chaperones versus the ability to fold and form a structure that's going to be in the virion on your own. So this we divide into self-assembly versus assisted. And we've already talked a little bit about this. There are many viral proteins that can assemble on their own. We've talked about the poliovirus protein, which as far as we know can assemble on its own. Who knows? Maybe a chaperone is needed and we simply haven't found it. Maybe they all need chaperones and we just don't know about them. But for now, we'll say some of them can self-assemble. And a couple more examples. HIV, the capsid proteins, if you express them alone, they can form empty shells. The influenza HA by itself, you just express the HA glycoprotein in cells, it will drive the formation of virus-like particles. And in fact, this is being done to make vaccines now, which have no nucleic acids in them. And finally, the HBV surface antigen, if you express that in cells, it makes virus-like particles. And in fact, that's the basis for the HBV vaccine. So these viral proteins have within them all the information, not only to fold properly, but to drive the formation of a particle. And in contrast, there are a number of viral proteins that can't assemble on their own. You sometimes need scaffolds, like we saw for the herpes viruses, or you need chaperones to form the structure. And as I said, this may in fact end up applying to even these instances. There may be chaperones that are involved that we just don't know about.